Welcome everyone to our first Q&A session for the Parkinson's Research Interest Group. And away we go.
So thanks, Hugh. It's a real pleasure to be here to do the first Q&A for this PRIG. And thank you to Martin Taylor for inviting me. I think he's not on the call quite yet. So yeah, my name is Tilo Kunath. I'm actually originally from Toronto. I did my PhD at the University of Toronto. I came to Edinburgh in 2003 to work on stem cells. And I've been working on Parkinson's disease, applying stem cells to tackle problems in Parkinson's disease for some time now. And I have a lab at the MRC Center for Regenerative Medicine.
Great, that's fantastic. Tilo, what is transplanting iPS stem cells? Are they stem cells, or are they dopamine neurons, or are they something in between?
Yeah, so that's a good question. So just, I guess, to give a brief introduction of what we mean as stem cells. So these are very special cells that are pluripotent, meaning that they can give rise to any tissue in the body. So we have the iPS cells that you've been referring to, induced pluripotent stem cells. And then the other type of cell that we use in the lab are embryonic stem cells.
Okay.
Both of these need to be pushed towards dopamine neurons, and essentially we push them towards a state that's we call a dopaminergic precursor before they're dopamine neurons, and it's the precursors that are useful and are efficacious for transplantation.
So you couldn't, can you take a dopamine neuron that's fully developed and transplant it?
So people have tried to take dopamine neurons that are, fairly mature, and essentially they don't survive. They're too mature, too fragile. Putting them into, at least in an animal model, they essentially is too aggressive a procedure. They don't like being moved after they've made their axons and dendrites. So it has to be a precursor, so a very small cell that's not quite made any branching yet. But it has to be on the route to making a dopaminergic neuron. There is this very fine window where you could transplant the cells that they're no longer stem cells, but they're not fully dopaminergic neurons yet. So
where do you insert these not stem cells, not yet dopamine cells, kind of halfway in between cells?
Yeah. So that's also a good question. So it's really specific. So where the placement of these cells occurs is exactly where the dopaminergic neurons innervate. So it's a part of the brain called the striatum, and the striatum can be split into two regions, the putamen and the caudate nucleus. Both of these regions are losing dopaminergic innervation. Essentially, the axons are dying off. So the precursors need to be inserted into this part of the brain to try to reconnect new dopaminergic terminals to the striatum. So the location is very, very specific that you need to place them, which is why you need very fine targeting by the neurosurgeons, the stereotactic setup, etc.
So they're not being put into the substantia nigra. They're being put into at the end of the trail of a dopamine neuron in the striatum, and then they're like sending out their tentacles and taking the neuron all the way back to the substantia nigra?
So I have to say that has been done in animal models, definitely. And I think there's one group, it was a Canadian group, Ivar Mendez, that did transplant into the putamen and into the substantia nigra. I think he's the only one that did it. I don't know how successful that was, I have to say, the human trials that were done in Halifax. But I know in the rat, if you transplant the dopamine precursors in the substantia nigra or into the striatum, there was functional rescue in the rat in either location. So it didn't matter if they transplanted cells into the substantia nigra or in the putamen. In the rat model, the rescue was the same. So moving forward, they chose to go for the putamen because surgically is much easier to access, it's less invasive. so I think for practical reasons, most groups chose to go for the putamen, except for Ivar Mendez, I think, chose to do deposition in both locations.
Okay, great. I'll ask one more question, and then I'm going to ask for questions from the group. The last question I'll ask for now at this time is, what types of subtypes, genotypes, or cellular mechanism types will a stem cell transplant likely work for, and which ones?
You mean people types?
Yes.
Yeah. So I think any condition where the substantia nigra neurons that innervate the striatum are lost, I think there could be very good use for a cell therapy that is dopaminergic neurons. So we published a review not too long ago in European Journal of Neuroscience that has just come out, that we argue that a really, really good patient subtype, or if you want to stratify patients, people that have lost only the dopaminergic neurons that innervate the striatum and don't have an active Lewy body type disease would be, we thought, ideal people to get a cell therapy, because their neuronal loss is extremely localized. They don't have all of the other non-motor symptoms that sporadic PD has, such as constipation and affecting the olfactory bulb or the cortex. So we thought these people with the Parkin mutation might be ideal. But I think sporadic PD, I think, is a great candidate as well, because there's lots of dopaminergic loss there. And there may be other conditions where the dopamine neurons that innervate the striatum are dying back. Those I think could also be candidates for a cell therapy.
Okay, I'm going to call for questions from the group.
If you inject, I'm talking in very simple words. If you inject the cells, do you also take out the dead cells, cells, to check what has happened to them? So when you put it really simply,
yeah,
when you put new ones in, you take the old ones out and have a look at them?
Yeah, that's a good question, and the answer is no. So they don't take any biopsies as they're depositing cells essentially in a very small syringe, and there is no mechanism to take a biopsy out. But I think when cells are dead and dying, they just get eaten up by surrounding cells such as microglia and macrophages, so that they essentially disappear. But you're right, like in glioma, you might want to take a biopsy and pull something out, but this is certainly to, the acronym cell replacement therapy, the CRT, is about putting cells in that are essentially lost. So it's not that the cells are there. Of course, there are neurons there that are not functioning well, but ideally you would want to just leave them there, because they may have some function. They may have, even if they're 50% functioning, you still don't want to get rid of those. So there's certainly no effort to remove any cells. Essentially, the cells that are lost have been lost over decades, and they're usually gone. Like there's nothing there to take out. But there may be some there that are not well. But again, I would also argue you should leave those there. Essentially, it's just a depositing of cells and then leaving the site with where you put the cells.
Great. Tilo, how is this going to work? Is it going to be levodopa first, then stem cells, and then once they're done with some DBS?
Like that. As you know, cell therapy is not a mainstream therapy yet, right? Maybe there's one, I would argue there's probably only one person, male in Japan, that has had the properly differentiated cells. So such early days, but it's a good question, and I spoke about it before, that we think that someone who's a good candidate for a cell therapy is usually in the more early stages of their Parkinson's. So so the trials that happened in the 80s and 90s, some of these individuals were quite late stage Parkinson's, and the cell therapy just didn't take. So there needs to be some innervation in the striatum for the cells to take, and the cells need to mature and innervate the striatum. So you set the timeline about right. You know, if and when cell therapy becomes a mainstream therapy for Parkinson's, you know, it would likely be offered early on. But after L-DOPA, like, you know, if you get diagnosed with Parkinson's, I can't imagine someone, even in the far future, saying, "Oh, you should get a cell therapy right away." L-DOPA, get on a list for cell replacement, maybe a drug that slows down the disease or halts the disease. But DBS is usually a much later type of therapy to control symptoms. And DBS and the now emerging, I don't know how much you know about focused ultrasound. That's being pioneered in Toronto by Andreas Lozano. Focused ultrasound and DBS I think are quite similar and would be used at a similar stage to control symptoms. And going back to the previous question, focused ultrasound, it doesn't remove cells, but it does get rid of cells. Focused ultrasound is a way to actually kill some cells. But these are not cells that are dopaminergic. These are not dopaminergic neurons, but these are cells that are causing, that are hyperactive and causing some of the tremor and some of the symptoms. So focused ultrasound does get rid of cells quite non-invasively, but they can't remove the cells. They sort of, essentially they cook some cells, and then the tremor can almost stop immediately. And then surrounding cells will get rid of that debris, such as microglia and macrophages will get rid of cellular debris. So there are some cases where you do want to remove some neurons, but they wouldn't be dopaminergic neurons, definitely not.
Oh. I'm omnipresent, Tilo. I'm everywhere you look.
Yeah. Hey, John, how are you? Good.
I'm good, mate. I'm good. Just thinking on the recipients of the cells, or the would-be recipients of the cells, is there any sort of age grouping that's been done to decide what would be the best age groups to do that, or is that still going to be coming from the trials in Japan and the US?
I think there is an age grouping. I don't think it's very restrictive for age. I think it was something like 40 to 70. But you know, it's known that younger people do better with invasive surgery. But I think the main criteria is the length of time or the advanced stage of the Parkinson's, how advanced it is. Because you could get, you know, Parkinson's at 65, and then at 69, you might be a great candidate for cell therapy. But if you got Parkinson's, if you had it for 20 years and it's, you know, and you're more a candidate for DBS, but you could be only 60, that, you know, you might not be included in or meet the criteria for cell therapy. So I think age has some part to it, but I think it's how advanced the Parkinson's is I think one of the major criteria that people will be looking for trials. Hi, Ben.
A quick question about iPS cells in general. They are obviously being used for a lot more than just cell replacement therapy. We're actually learning a lot from the use of these cells. Could you talk a little bit about how it's actually advanced just our basic understanding of this disease?
Yes, absolutely. So iPS cells, in addition to being useful for cell therapy, we use them to model disease a lot. So iPS cells, when you make them, you can preserve the genetics of the person that you made them from. So this is very useful if you want to study a genetic form of Parkinson's that looks like a sporadic form. And in my case, we used iPS cells from a family that had a triplication of alpha-synuclein gene. So, you know, I think they're wonderful for disease modeling and for testing drugs. It's not easy, it's challenging, but iPS cells have been made with LRRK2 mutations, with GBA mutations, also PINK1, Parkin mutations. There's a lot of work going on trying to understand Parkinson's, the mechanisms, but also to test drugs on these neurons. And, you know, I think this is where the big impact will happen. You know, when there's drugs that slow down Parkinson's that could be discovered with iPS cells, that's going to be for the vast majority of people. We didn't get into who cell therapy might be for, but, you know, I think it'll likely be for a small, well, not necessarily small, but it's going to be for a subset of people with Parkinson's. But a drug that slows down the condition, this could be a game changer, and a lot of people could be on that. And iPS cells could be used to discover such drugs.
The Parkinson's disease. Which other diseases could stem cell therapy be applicable to? Which other neurological diseases could it be applicable to?
Yeah, that's a good question. So there is this, I think the second biggest effort. Well, actually, the other disease that it is going to clinical trials, but not a neurological condition, is age-related macular degeneration. So
yeah.
they're making retinal pigmented epithelium in a dish, and they're transplanting into the eye. Huntington's disease, there's some efforts towards cell therapy for Huntington's disease. They're also, which look fairly good. They're making striatal neurons and transplanting those in animal models, I should say. Spinal cord injury has been, they've been trying to get something going for spinal cord injury. It doesn't look fantastic at the moment, but who knows? Yeah, there's other conditions that cardiac. So cardiac, people are trying to make cardiac tissue from iPS cells or ES cells to transplant. But a lot of these conditions outside of the brain, they're having difficulty getting the cells to the right level of maturity. So you need the cells to be quite adult-like. And I usually say this in talks that Parkinson's disease is low-hanging fruit for cell therapy, because the tissue that you need to make in the lab is fetal tissue to transplant. And ES and iPS cells make fetal tissue quite well, but it takes a long time to make adult tissue in the dish. But if you want to transplant cells with cardiac properties of an adult, it will take months, maybe a year of culture in a dish. But for Parkinson's, because the very first question that I got from you is that the cells can't be too mature. If they're too mature, they won't transplant. So you actually need cells with fetal properties for transplantation, which is, again, why I think Parkinson's is the first major disease that these cells will be used for cell therapy.
What about MSA and PST?
Yeah, MSA is a good question. So MSA is more a disorder of a different cell type. It's called an oligodendrocyte. So these oligodendrocytes wrap and insulate neurons. Transplanting oligodendrocytes is not, making them, first of all, is not trivial, but transplanting them is also, it's not clear that will work in models. So there's no good animal data to say that will work. But in theory, it could be used to try to fix the substantia nigra innervation of the striatum, which is affected in MSA. But one of the big issues with MSA is it's quite aggressive and quite fast. So we published another paper in November where we made neurons that were resistant to getting synucleinopathy or Lewy bodies, or essentially Parkinson's-like disease. So I would argue that these might be the more useful for MSA. But again, these cells will only fix the dopaminergic cells that are lost in MSA. The oligodendrocytes that are affected, this cell therapy won't help. So this is a trickier condition to treat with cell therapy, but it's, it could be beneficial, but it's not one of the first conditions that it would be tried, I think, for cell therapy. For drugs that slow down alpha-synucleinopathy, I've been discussing with people in Edinburgh that MSA patients should be the first people to get drugs that are opposed to slow synucleinopathy, because the synucleinopathy in MSA is quite rapid and quite aggressive. So if you want to see an effect from a drug, you might need a 10-year trial to look at Parkinson's, but in MSA you could see effects much quicker. So PSP, I don't know. I'd have to get back to you on that one.
Okay. Yeah.
This is Bill Curtis. I'm sorry I'm late, but I guess my question is on these cells that you differentiate and cause to revert and then come back, how do you know that they are behaving like the neurons would in their environment? I guess there's different cells that you can use for current testing that aren't these cells, and are there differences in the induced pluripotent or whatever IPA cells that IPS cells that you don't find in these other strains of neuron models?
So there's a lot of different stem cell-like cells out there. There's mesenchymal stem cells that can be derived from bone marrow. There's cells that can be derived from fat, dental pulp as well. So at the beginning, I was talking about pluripotent stem cells. And in my opinion, and in many people's opinion, scientists' opinion, these are the only types of cells that can make true dopaminergic precursors. So they're either IPS cells, as you were describing, or they're a cell type that was discovered a bit longer ago in 1998 called embryonic stem cells. These embryonic stem cells come from a blastocyst, that's a small round embryo from an IVF clinic. So these are the two types of cells, IPS, and the other one is acronym is ES, that can make true dopaminergic precursors that will function in lots of models, including rat models, mouse models, monkey models, and soon we hope in humans. There's no other cell type that can do that. There's no other type of stem cell that can do that. Mesenchymal stem cells can't do that. There's other type of neural stem cells that people grow, and those can't do that. So in my opinion, there's only this one type of cell, even though they have two names, you have IPS and ES, that can make the true dopaminergic neurons.
Yeah, I guess my question was, researcher was contemplating whether to buy the kit to make the stem cells, and I guess you can revert any cell back to a stem cell and then go forward.
Yeah.
Or
I see what you're saying. Where do you...
And the question became, how do you know which one is behaving more like the natural neurons?
Yeah. So IPS, so IPS you can make from skin. IPS cells you can make from blood. You know, people have made it from hair follicles, even any bit of tissue, you know, that you could get, you could in theory make IPS cells from. And there has been some debate as like, oh, if you make it from skin, it's better than blood, et cetera, et cetera. But, you know, all that is proven to be not nonsense. It doesn't matter where you make the IPS cell from. If the IPS cell is made carefully, and it's proven to be a fully reprogrammed IPS cell, it will behave like an ES cell, or it'll behave the same if it comes from blood or from skin. There's this thing called epigenetic memory that may exist for a few months for the life of that IPS cell, but essentially that, you know, those epigenetic marks get erased and the cell becomes truly pluripotent. So it doesn't matter which part of the body you derive IPS cells from. And now, you know, I think a lot of people are deriving it from blood because this is the least invasive type of cell to donate. So cells that I made IPS cells from were skin. And, you know, the people that donated these skin had a half centimeter disc taken out of their forearm, and they had to have a couple stitches. Takes a couple of weeks to heal, you know, so it's not so pleasant. So a few mils of blood is much better, and this is now the way that most IPS cells are made, and they will work. They'll work just fine. And you can buy kits to make these IPS, but, you know, I've made a lot of IPS cells in my lab, and the quality of them can be very variable. So you need to just spend a lot of time to find the IPS cell line that is a good one. The patient that we made IPS cells from in Iowa, we made 40 independent lines. Sorry, 30. We made 30 independent lines from this patient. We made 10 lines from her daughter. And they weren't all the same. These lines
Tilo, if I may, I think the question that William's asking is that after you differentiate them into dopamine-producing neurons, how similar are they to real dopamine-producing neurons, or quote-unquote real dopamine-producing neurons? And how tricky is it to get them to kind of grow in their quote-unquote native environment?
Yeah, that's all has to be tested empirically. And they are as similar to real neurons derived from say an embryo as possible. So it's been published that when you compare fetal tissue dopaminergic neurons and cell-derived dopaminergic neurons, their behavior in an animal model, they look essentially the same.
Yes.
So
good.
They can't, I don't think they'll be completely identical, but they're very, very similar. But what's happened to these neurons is that you're asking them to start, from a fetal stage and grow inside an adult brain. So it's certainly, it's not natural. It's a bit, so you're asking very, maybe, equivalent to 6 to 9 week old fetal neurons to function in a 30, 40, 50, 60-year-old brain.
Right.
But I think those neurons, if you transplanted fetal tissue, like true human fetal tissue, or lab-derived neurons or neural precursors, their behavior is functionally really, really similar.
So Tilo, what are the risks involved in stem cell transplants?
There is risks, of course, for neurosurgery. Neurosurgery is associated with infection risk, hemorrhaging risk. I think they're not huge. Of course, they're definitely not huge, but the numbers are, I can't give you exactly. A neurosurgeon would be able to give that to you. So the risk that most people are worried about when you're transplanting cells is, will some of them grow out of control and create a tumor, right? So that's risk not associated with the surgery itself, but with the cells that you're putting in. And I can say that, since 2011, when this type of cells were being made in the lab, hundreds of mice and rats and monkeys have been transplanted, and there's no evidence of tumor formation if the cells are made in the correct way, in the way that was published by Lorenz Studer in 2011. But there's always a risk. So what is happening is that there's a large batch of cells being produced. They're all frozen. And then there's a lot of animals that are transplanted with these cells to see if there's any tumorigenic potential in these cells at all. And all these safety tests have to be done before you can release cells for cell therapy. So Jun Takahashi published in Nature not that long ago. He had iPS-derived neural precursors in monkeys for 2 years, and there was no evidence of any overgrowth or any tumor formation. So, yeah, this all has to be mitigated. It all has to be tested, and every time you do a differentiation, you could have you will have a different batch of cells. So the cells that are differentiated, that batch that you made in January 2019, whatever, that particular batch needs to be tested for tumorigenicity. And then if it shows that there's no tumorigenicity, then you would, quote, you'd release that batch for a therapy.
Do you think is going to be attributable to just adding a cell that can produce dopamine versus adding a cell that has the right axonal branching to make the proper connections? Yeah.
So just making dopamine isn't enough. So I think there's about 9 different types of neurons in the brain that make dopamine. And it has to be a dopaminergic neuron with this midbrain substantia nigra-like properties. And this is getting a bit outside of what I know, but there's certain properties for these neurons that have pulsatile release of dopamine. Mm-hmm. So the pairing of this type of dopamine neuron has to be very specific. It can't be just any dopamine producing neuron. So there have been trials of adrenal medulla being transplanted because it makes dopamine. So it's not just simply having a dopamine pumping neuron in there. It has to be of a certain physiological and electrophysiological phenotype. So we're very, very work very hard to get a substantia nigra-like dopaminergic neuron. It can't be any type of dopaminergic neuron. This is to do with the electrophysiology to connect with the striatal neurons.
Yep. Hi, so Claire Lehman here. You mentioned earlier, Tilo, about the sort of subset of patients that might be most sort of amenable to having stem cell therapy, which I was wondering if you could define that a little bit more.
Yeah, so we wrote an opinion piece recently. It's open access, and it was titled "Are Parkin Patients Ideal Candidates for Cell Replacement Therapy?" Do you know what Parkin gene is?
Yeah, I've heard of it.
Yeah. So it's a gene, it's an E3 ubiquitin ligase, which means nothing, sorry, but it's a gene important for mitochondrial function, and it was discovered in Japan. And this, if you have two bad copies of Parkin, you essentially present with Parkinson's. You have Parkinson's symptoms, you're levodopa responsive, levodopa helps a lot. These patients are usually young onset, but they're very slow progressing, and they're levodopa responsive for 20, 30, 40 years. And almost none of them go on to get dementia, or none of them I see go on to get dementia. And but some of them have died recently. Well, not recently, but since the 1980s, some Japanese families with this Parkin mutation, before they even knew they had a Parkin mutation, came to autopsy, and a lot of them don't have Lewy bodies. The pathological hallmark of Parkinson's, they don't have Lewy bodies. And then also, they have very little neurodegeneration anywhere else in their brain. The locus ceruleus has some neurodegeneration in some of these patients, but it's very localized. So we argued that Parkin patients, because they have very localized substantia nigra loss and they don't have active synucleinopathy, that these would be, we thought, ideal candidates for cell therapy. Because the cells that we make in the lab are, the nigral dopaminergic neurons. You transplant them in the putamen of a Parkin patient. And we know now that transplants, they get Parkinson's eventually. It takes a long time, but they do get Parkinson's because of what we think is synucleinopathy going into the graft. But there's no synucleinopathy in Parkin patients, so we thought, okay, the graft will not succumb to a Parkin disease because Parkin sits inside the mitochondria, and the cells that were transplanted will have a normal Parkin protein and normal Parkin function, normal mitochondria. So we argue that these would be, ideal candidates. But, I think Parkinson's disease itself, sporadic PD, I think is a great candidate as well for cell therapy. And it could improve a lot of the symptoms associated with a dopamine loss, loss of dopaminergic innervation. And, we write also that these cells won't contribute to any symptoms that are not due to dopamine, won't help symptoms not due to dopamine loss. So a lot of the non-motor symptoms these cells will not help with.
Okay, so says more specifically that a genotype would be useful and advantageous to help select the patients than just a phenotype.
Yeah, you can do some phenotyping as well. So there's ways to see if you have an active synucleinopathy. It's not easy. So we point out some ways in the paper. There's a cerebrospinal fluid test that is done at a research level. Is there synuclein oligomers in CSF? Because some LRRK2 patients, I think, definitely have come to autopsy and don't have any Lewy bodies. Okay. The LRRK2 is a much more common mutation. It's autosomal dominant, so we need one mutant copy. But I've heard it argued that half of LRRK2 patients don't have Lewy bodies. I'm not sure if it's that high, but certainly something unusual is going on there, that their Parkinson's may not have anything to do with alpha-synuclein. So there may be other groups. So there's two other genes, PINK1 and DJ1. It looks like these people with PINK1, DJ1 mutations also don't have Lewy bodies, and they have a very localized substantia nigra loss and no other pathology. So they might be ideal candidates as well. So some phenotyping could be used to find people that have very localized nigral loss and no synucleinopathy. I think they would be ideal candidates.
And in terms of clinically identifying that cohort, how do you go about that?
Yeah, it's not easy. Genetics, of course, is the easiest. If you're in a Parkin family, then that's of course the easiest. But there are some people with Parkin mutations that do have Lewy bodies, especially heterozygous mutations. So we argued even if you have a Parkin mutation, you should still check non-invasively or some way, is there synucleinopathy active? But screening, I don't think it's an easy way to find them. But genetics is the first, with all the genetics that can be done now, genetics is now much, much, much quicker. If you can find
we waited for a year.
Well, yeah, in the lab it's much quicker. Where have you been waiting?
I'm in the UK, so yeah, Professor Morris took my sample a year ago, but I'm still waiting for the results.
Yeah, no, I know the story. Yeah, are you UCL?
Yeah.
Yeah, yeah. Keep knocking. You know, Hugh Morris. You knock on Hugh Morris's door.
Yeah. He's been excellent. To be fair, I'm not whinging. He's been amazing, and we have a great service. So like, I'm not complaining. But yeah, it's a little bit frustrating because you think, oh, is it gonna? Sorry, my hand's shaking. Sorry. Yeah, is it going to kind of when yeah, it will have a bit of an impact now, so it'll be useful to find out. But no, it's useful to, yeah, I'll keep waiting.
Or keep yeah, no, he was great, but yeah, you know, these things take time, but yeah, it is frustrating. I can you know, in Scotland as well, we have Mariette in Dundee who's doing a lot of the genetics, but yeah, keep knocking and keep advocating and push.
I'm being polite. Assertively polite.
Thanks. I have a couple of questions, actually. So first, you were talking about creating cell lines from that one patient and from her daughter. So I'm wondering, when you initially started talking, I thought you were creating the stem cells from each individual patient to use in themselves, but that sounds like maybe not the case.
No.
So you're implanting from a line that was created from one person into everybody.
Yes. Yeah. So the idea is to get one line from a person that, of course, doesn't have any Parkinson's mutation, so one donor, and that single donor line would be used for everyone. So the characteristics of that line will be very well known. You could, the cells, I didn't mention at the beginning, but the cells are immortal, so you can make huge volumes of them, you know, billions of cells, and that one genetics would be used then for everyone. And there would be some immunosuppression, as you would imagine, because these are, of course, foreign. But the immunosuppression doesn't have to be a lifetime because the brain is immune privileged. So I think the trials will use immunosuppression for 1 year, and then the patients will come off the immunosuppression. But definitely this personalized, there was an idea of personalized therapy. You have your own iPS cells made into dopaminergic neurons and transplanted back. That's a non-starter right now. That's not going to happen.
And then you were talking about functional rescue, and I realize we haven't done trials in humans yet, so this may be a hard question to answer, but I'm wondering like how complete the functional rescue is and how long it takes to reach full efficacy and how long it...
Well, there has been cell replacement therapy has been done for Parkinson's since 1987.
So do we have a sense of like if you get a stem cell
yeah, there is a sense of how long it will take and if it works, what will happen and how long it will work. So the first point is it's not quick. So when fetal cells were transplanted in 87 and onwards, to see real benefit was more than 1 year. 2 years and 2 to 3 years, if it was going to work, is how long it took to see some benefit. So it's simply a time for the cells to mature and reinnervate. So they have to be transplanted as precursors, and then you have to give them time to mature. So, you know, some of the trials endpoint was 1 year, and it was found out that's too early. So we know that if they do work, they will take, you know, 2 to 3 years to really mature. And we also know that fetal grafts, when they did work in some people, they worked for a set amount of time and then began to fail. So there's some patients that had some relief for maybe 10 years, 15 years, reduction of some symptoms, reduction of L-DOPA even, but then symptoms started coming back. And then also what was happening was that some of the patients were progressing to dementia, which these cells have no protection against dementia. So the synucleinopathy is not slowed down. So the spreading of the Lewy bodies is not slowed down by these cells. They go into the cells that are transplanted. They go to other parts of the brain that the cells don't contribute to. So there is a window where if it works, it will take some time. It could work for a decade. It could work for a decade and a half. But then the Parkinson's isn't stopped. The Parkinson's will then go into the graft. So, you know, we need drugs to stop the condition. right? So an ideal therapy would be replace the dopaminergic neurons and stop the spreading of the disease. We've also published disease-resistant cells, where the cells we think will not get the condition, but that's again not going to stop the synucleinopathy from spreading to other parts of the brain. So the dementia aspect, even with disease-resistant cells, can't address that. So yeah, we do know, we have some expectations of what will happen if it does work, based on fetal grafts that have been, there's about 300 people that received a fetal graft since 1987. And some of them, no one's asked me yet about graft-induced dyskinesias, but one of the major side effects of the fetal grafts was something called graft-induced dyskinesias, which were very, very, very troublesome. And this is probably what stopped all of the cell therapy. That may have been due, well, we think it was partly due to contamination of serotonergic neurons, but not entirely. So I think this is one of the biggest worries of people doing cell replacement therapy is, will we see graft-induced dyskinesias again? We don't know yet, because there's barely anyone transplanted yet. But what we do know is that there'll be no serotonergic neurons transplanted. So the issue of contaminating serotonin neurons is solved when you grow them in the lab. But if that wasn't the entire cause of graft-induced dyskinesias, we may see them again. And then how do you combat against that? How do you deal with that? It's just something that people are going to have to figure out as we move forward. Yeah, so that's just, for me, I think this is one of the bigger worries, that the cell therapy may crop up. I don't think there's going to be tumors or there's going to be any overgrowth, but it's the graft-induced dyskinesias that could be the most problematic.
Even though the disease is still progressing while you're doing the implant, and then it takes 2 to 3 years to mature, like how much relief might somebody get for those 10 years? Like it's almost like they're running a race. Cells are running a race against what's still going on in your brain. So you tend to get like a 30% reduction or like 100% reduction, or how?
Well, I've seen some amazing videos of people that are look almost normal. So I had a student working with me, and he went back to UCL, and they brought in a patient to assess, all the students to assess this patient. And all the students were asking to do a number of things, number of tests, and they just like gave up. They're like, I don't know, we don't know what's wrong with this guy. And they said he's got Parkinson's disease for 15 years. Wow. They had a graft 5 years ago, something like that. And his symptoms were so non-existent that a set of medical students couldn't figure out what was wrong with him. And I've seen a video from Kurt Fried, in Colorado, show a woman that could barely get up. This is before her medication in the morning, barely get up from one chair to another. And 1 year after graft, she was up in one chair, moving to the other. And then she showed a video 4 years after graft for playing tennis, right? So I was like, but these are of course extreme cases of people doing well. But I think if it works well, there's, there could be very significant reversal of symptoms for a finite amount of time. But, that's why we wrote this Parkin article. We think Parkin patients, if you fix the substantia nigra, the nigral innervation, and they don't have a spreading Parkinson's disease, such as synucleinopathy, a Parkin patient may have a lifetime reversal of symptoms, right? So this is why I think they should be first in the queue, because they're the ones I think that will benefit the absolute most with a cell therapy. Their cell therapy will unlikely to succumb to an ongoing disease. And, they could be reversed for their lifetime.
That's cool. So my last question is just how far are we from more clinical trials and from this actually being a viable treatment for people?
So Japan has already started in October of last year. And I think Lorenz Suter may announce any day, any week now that he's also started. So Lorenz's trial was supposed to start in December of last year. But I think there's some regulatory hiccups, but I think it's imminent that he will transplant the first patient. I think the first 10 patients are already chosen. I know he produced the cells in 2016. So the cell batch has been produced several years ago, and they're frozen. and they've gone through all the efficacy and safety testing, all passed, and they're just getting final paperwork hurdles done with the FDA, I guess. But I know he wanted to be in first patient in December, but I think it'd be imminent that he's in a patient, as first of 10 very soon. But certainly it's already started in Japan. So if you go to, there's a Nature News article that the first 55-year-old patient has had their surgery, and they'll do their second soon, I presume. I think they're going to go up to 12 patients in the next year or so. So the answer is it's already started.
Cool.
Hi, Tilo.
Hi.
I just wondered, how far off are you off developing the cells that are resistant to further spread?
Yeah, that's that was the.
Is there any downside to having cells that are resistant to death?
Well, they're not resistant to death. They're definitely not resistant to death.
Okay.
We think they're resistant to getting Lewy bodies.
Right.
Yeah. So and yes, we think there might be. We talk about it at the end. What's the maybe negative of deleting? Essentially, we deleted the alpha-synuclein gene, which makes a protein needed to make Lewy bodies. So so one argument that people say that the dopamine release will be not right. The release of the dopamine won't be normal. And there's some papers that say there might be hyper release of dopamine, which may sound good, but it may not be good because it may be uncontrolled. So that could be one negative thing. But the thing that I think maybe more, well, not more concerning, but I think will be an issue is that mice that don't have synuclein, they perfectly normal, most mice, if you look at a mouse that doesn't have this gene, they seem quite normal, unless you infect them with a virus such as West Nile virus or a virus that invades the nervous system. So mice that have lost this alpha-synuclein gene are very susceptible to West Nile virus. They die very, very quickly. So this is part of an emerging area that synuclein is not important for synapse function, but is actually important for protecting against viruses. So if you delete synuclein from neurons, now these neurons, yes, they don't get Lewy bodies, but they may now be susceptible to get virus and may not be able to protect themselves. So you'll be interested in this. So the person that published this, his name is David Beckham, and he has a lab in Denver. So we have a collaboration that's just initiated. So he's published the synuclein null mice are very susceptible to West Nile virus. So we're going to test the neurons that we've made. We're going to see how susceptible our human neurons are to West Nile virus. And the prediction is that they will be quite susceptible to West Nile virus. So that's I think one downside of having deleted this gene, that they may be now, if an infection occurs, they may be very susceptible and they may die very quickly, rather than if you have synuclein, they'll protect themselves. So David Beckham also published that if you delete one copy of synuclein, if you just delete one, the mice are actually just as able to fight off West Nile virus as if you had two. But in our cells, when we delete one copy of synuclein, their speed of getting Lewy bodies was much, much slower. So as one sort of in between halfway house is that we could make cells that have lost one copy but not two copies of synuclein.
See, and does that, that's kind of suggests that the role of alpha-synuclein is to do with viruses.
Well, this is why people think it may have evolved for that, is to protect against invading viruses in the nervous system. It evolved during bony fish development, and our immune system has constantly been improving and evolving. And one of the ideas is that synuclein, its function is to protect against viruses.
Mm-hmm. Mm. Yeah, you mentioned that there were some of these patients who showed extreme relief from Parkinson's symptoms. Do you know anything about their genetic makeup? Have they been specifically tested for DJ1, PINK1, or Parkin?
Yeah, I don't think they have, but I think it's very unlikely that they had these genetic forms of Parkinson's. They seemed at autopsy like typical sporadic PD, meaning they had Lewy bodies, they had the pattern of degeneration that a sporadic PD has. Yeah, I think, yeah, I'm sure almost none of them were genotyped, but my impression from patients that had come to autopsy is that they look like sporadic PD.
Okay.
Last question I'd like to ask Tilo before I go. When you look into the far future of cell replacement therapy, what is it that you see? How is this going to be applied, not necessarily just to Parkinson's disease, but what are the kind of upper limits of this technology, say 20, 30, even 50 years down the road?
So the far future of cell replacement therapy is it's not going to be needed. This is my view. My view of cell replacement therapy is going to have a window of usefulness, and I think in 50 years, for Parkinson's is not going to be needed. Absolutely. So I think I've told you my view of how Parkinson's is going to be dealt with. Parkinson's needs to be dealt with early detection, and then some sort of drug therapy to prevent the onset of the condition. So all these drugs that we're talking about that you could find with iPS cells. So I think Parkinson's needs to be tackled before dopaminergic neuron loss becomes major. This is my opinion. So if you can detect someone before they even have any motor symptoms and they can prevent the motor symptoms from onsetting, then you're not going to need a cell replacement therapy. But also, I think in the future, if you really want to get futuristic, I think cell replacement therapy will be replaced by something called direct programming, where we use viruses to change endogenous cells such as astrocytes into dopaminergic neurons. And we're actually working on a project like that now. There's many labs working on this, but I think transplanting cells will have a finite lifetime for medicine, and then they'll go away. There'll be no need for it, either because you can detect Parkinson's so early and prevent it, or viruses will be used to convert endogenous cells into neurons. And that is very useful because in that way, you don't need cells from a different genetics. You have your own cells converted, and it doesn't go through the process of making iPS cells, making them into dopaminergic neurons and transplanting. You're just instructing cells in situ to say, stop being a microglia or stop being an astrocyte and become a dopaminergic neuron. So either... Yeah, so those are the two things. Either there's going to be no need for it because we should be able to catch Parkinson's before there's such neuronal loss, but then they never get to a stage where they need a cell therapy, or the sophistication of the methods becomes such that instead of using cells, we use viruses to instruct resident cells to become dopaminergic neurons instead of transplanting dopaminergic neurons. So this is the future. But it might be 50 years from now, unfortunately. So cell therapy definitely has an important window right now. There's lots of people, I think, that could benefit from a cell therapy that have Parkinson's now, that will be getting Parkinson's in 5 years, 10 years. You know, if you can replace a broken part or lost part, you know, it's essentially lost parts. If you can replace it and restore function, you know, we have to go for it.
So first of all, thank you, Tilo. Second, is there anything you would like to ask us?
Oh.
Is there anything you would like to ask us or like us to do? Or I'm a Parkinson's patient, I'm in the advocacy group. Is there anything from you asking us? Or
well, I think it'd be ideal to spread education, right? You know, I try to do what I can, but you know, I think this is brilliant for disseminating information and dispelling myths about what a cell therapy is. Yeah, you know, I think this PRIG is fantastic. So I hope, so the video that I sent through, I'd also like that distributed on the Facebook side if possible. I think spreading information and advocating for this type of therapy, I think it would be fantastic. And also, you know, I can't remember her name, but the one at UCL, you know, pushing and being polite is of course good, but asking for more services in Parkinson's, I think is essential, because, you know, I think from a research perspective, you see how much can be done, but then, you know, in the constraints of a healthcare system, such as free healthcare systems, such as in Canada and the US, is always difficult of what will be done because of finances. But you know, from the Parkinson's perspective, we need to keep pushing at the door, keep advocating, keep getting better and better services.
Thank you so much, Tilo. That's just fantastic. Big round of applause, and we really appreciate you spending a whole hour with us today. Thank you for joining us today. Hope you've enjoyed this session for the Parkinson's Research Interest Group with Tilo Kunath on stem cell research for Parkinson's. Have a great day.